Today we will discuss the final video about the paper where we are comparing the effect of DANA. Not comparing, we are analyzing the effect of DANA on different aspects of the cell. So far we have understood that DANA has a effect on the sialic acids that are expressed on the cell surface. That is, it acts as a sialidase. At the same time, it does not have any other effect on the cell surface so far. Some of these effects has been observed, and some of these effects such as CA2 oscillations, sialic acids expressions, and its comparisons with Sialidase isoenzyme Neu3 will be discussed in this video. And this effect will be predominant on the insulin when there is a minimum threshold of glucose are present, that we have discussed in the previous videos. So let us continue the topic of this video, or let us start the topic of this video, that is effect of DANA on Ca2 plus oscillations. So the panel A or the figure A on the top left hand side will represent the effect of DANA on Ca2 plus oscillations, and here we can see that the glucose levels are minimum, that is around 2.8 millimolars. They do not see too much oscillations of the Ca2 plus. However, when the glucose levels are around 8.3 millimolars, one can see the Ca2 plus oscillations. But when DANA is injected, they do not see any sudden spike or change in the glucose levels. And these things are been quantified in the left hand side, where we can see that the frequencies of this glucose with 8.3 millimolars and glucose with 8.3 millimolar and DANA does not get changes, and same results are observed for the amplitude also. The amplitude does not get changes in the conditions where only glucose are present at 8.3 millimolar or glucose with 8.3 molar and DANA is present, indicating that this DANA does not have any impact on the Ca2 plus oscillations. However, for the positive control, what they did is even in this 2.8 millimolars with the glucose, they do not see any effect, and when the DANA is present at 2.8 millimolar, they also do not see any effect. But when the control, that is TB, is used, they can see a sudden spike in the oscillations of this glucose, oscillation of this Ca2 plus, not glucose, this Ca2 plus oscillations. We can see that sudden spike whenever this positive controls TB is used, which affect somewhere affect the phosphate group. I'm not able to recall the exact name, but this is a positive control, so indicating that the system is working properly, but they do not have any changes in the Ca2 plus oscillations when DANA is used. And when they check on the sialic acids, what they find out is that in the case of the negative control, that is PBS, they do not see any expressions of the sialic acids on the cell surface. And this one is the DAPI, and this is the FITC-STNE. And when they use the DANA, they can see that sialic acids expressions are more in the case of DANA. That can be seen here, that in the case of DANA, the FITC signals are more predominantly stronger compared to the PBS. And the quantifications are shown in this right hand side, where we can see that in the case of control where PBS is used in the exocrine tissue, they see a control level signals, and in the pancreatic islet they also see the control level signals. However, when the DANA is used, signals of this FITC is statistically significant, or it's higher compared to the control case where exocrine tissues are being analyzed. So in the pancreatic islet, when the DANA is used, they see the more sialic acids formations. And after that, they compare this compare the DANA effect on glucose and blood insulin with this Sialidase isoenzyme Neu3, which is naturally found in the mice. So what they have can do is they knock down Neu3 knockout and check the glucose level, and they see that the Neu3 knockout the glucose levels are lower compared to the wild type. However, and similarly, they also check the insulin levels. Those they can find out that insulin levels are higher when the in the case of Neu3 knockout. So this indicates that the results of the Neu3 knockout plays a similar role that Dana is playing artificially. After that, in the fasting condition, is fasting for the 24 hours, they do not see too much changes. They do not see any statistical significant changes between wild type and Neu3 knockout cells. But when glucose are introduced, they are able to see the and when glucose are introduced they are able to see, and also this Neu3 siRNA are introduced, so they are able to see the increase in the insulins that can be seen at this results uh, after the glucose injections and also this Neu3 siRNA injections. So they can see that insulin levels are statistically significantly higher compared to the case where negative siRNA is used. So when siRNA, that is, when the sialidase enzymes increases, it will increase the insulin levels, provided the glucose are present above a minimum threshold. In this case, around 8.3 millimolars. And in the panel, in the effect of the Dana on the sialic acids, that is on the bottom left figure, the observance are of the Dana and PBS are done 30 minutes after the glucose injection. So first they inject the Dana and thereafter they inject the glucose, and 30 minutes after that they observe this uh, observe the sialic acids. So I hope you understood how the Dana is effect of Dana on the blood glucose and insulin. However, it does not affect other main characteristics that are been analyzed in this paper. So if you need to understand more about this uh, effect of Dana or these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.